In this lecture, we're going to talk about patterns of inheritance, how we as humans inherit our traits, and in particular, how we inherit different disorders. I think most people find this topic pretty interesting. It was first figured out by an Austrian monk named Gregor Mendel. So this is often called Mendelian genetics. We'll look at Mendelian genetics in this unit, and then in the next unit we'll look at what's called molecular genetics, how we're really, from a molecular standpoint, inheriting those traits. So let's look again at a human karyotype, and just remind everyone that we have 2 of each chromosome. So we have 46 chromosomes as humans, 23 pairs. We got 23 from the egg that made us, and 23 from the sperm that made us. And that ended up producing what we call a zygote with 46 chromosomes, 2 of each. So we got 1 full set from mom and 1 full set from dad, if everything went well. It's important also to note that if something goes wrong during the production of the egg and sperm, and this chromosome number is not correct, or a bad sequence of DNA is inherited, this original zygote is going to divide to produce every cell in your whole body. So if there's a mistake here, then every cell of your whole body has that mistake. This is a big deal. If something happens when your skin cells are dividing, something goes wrong during mitosis, no big deal. It'll be kicked out of the cell cycle at some point, hopefully. But if a mistake happens in meiosis, or if a defective gene is passed on to this egg or sperm, that entire organism is going to inherit that. So this is a very important topic. Remember that chromosomes 1 through 22 are called the autosomes. We've seen this term a few times now. And 23, those are the sex chromosomes. So this individual has 2 X's, so that would be a normal female. So let's start looking at the patterns of how we inherit things. And in order to do that, we need to define a few terms. One of the terms we need to understand is a very important term called allele. What is an allele in genetics, in heredity? An allele is another way of saying an alternate version of a gene. So let's pretend that this is a chromosome. Let's call it randomly chromosome 18. And on this chromosome, this is a hamster, and on this hamster's chromosome 18, this is where fur color is determined. And this hamster has 2 possible fur colors, brown and white. In that case, we can have 2 different alleles here, 2 different versions of that same gene. We could have the brown fur allele, or we could have the white fur allele. Those are our 2 possible choices. So alleles are alternate versions of the same gene. What is a gene? We've talked about this term before, but let's just remind ourselves. A gene is a region of DNA that codes for 1 trait or 1 protein. So gene, a region of DNA that codes for 1 trait. one protein. It's really a sequence of DNA located in a specific region on each chromosome. We know that we have what are called dominant alleles and what are called recessive alleles. So those terms are very important also in understanding inheritance. So dominant alleles and recessive alleles. And we'll further define that as we go along. We also know that since we get a chromosome from mom and a chromosome from dad, we have two alleles. So this hamster got one chromosome 18 from mom and one chromosome 18 from dad. And he's going to have that fur allele in the same location on both of those chromosomes. And he has three possibilities. Maybe he inherited brown fur allele from both parents. Maybe he inherited one of each, brown from one parent and white from the other. Or maybe he inherited two white fur alleles. Those would be the three possibilities. So he either has two alleles that are the same, or he has one of each. So if you have two alleles the same on your two chromosomes, you're called homozygous for that trait. This term zygous goes back to that original cell that divided to make all of your cells, which was called a zygote. So you either got two the same, or you got two different alleles. And if you got two different alleles for that trait, you are what's called heterozygous. Homo means same and hetero means different. Heterozygous means you got two different alleles for that trait. If you're heterozygous, we don't need to say dominant or recessive. It means you got one of each. So in this story, it would mean you got a brown allele from one parent and a white allele from the other for fur color. Homozygous, if you're homozygous for fur color, you either have two browns or two whites. And in that case, we do need to use another term to differentiate so we know which one you got both of. So you are either homozygous for the dominant allele or you're homozygous for the recessive allele. So in the world of homozygous alleles, you're either homozygous dominant, so that means you got two of the dominant allele, whichever it is, or you're homozygous recessive. And we call this an organism's genotype for that trait. Genotype is what two alleles you have for that trait. Are you homozygous dominant? Are you homozygous recessive? Or are you heterozygous? Those three terms represent your genotype. Your phenotype is what do you look like as a result of that genotype? What is the physical expression of that genotype? So phenotype is physical expression of your genotype. Do you have the disease or do you not? Are you brown furred or white furred as a result of that genotype? So these are important terms as we go through this story. To determine what's dominant and what's recessive depends on the type of inheritance we're talking about. But usually what we're doing in all cases in determining what type of inheritance pattern we're looking at is it's significant to look at the heterozygous individual. the heterozygous individual is going to tell us a lot about what kind of inheritance we're talking about. If we're talking about the fur color and the genotype of that hamster was that he had one brown allele and one white allele. In other words, he was heterozygous. And his phenotype was that he's brown. We would know that brown is the dominant allele. And we would know that we're talking about a case of what we call complete dominance. And that's the first pattern of inheritance we're going to look at. We're going to look at a pattern of inheritance in which one allele is completely dominant over the other allele. So in this story, it's similar. It's rabbits instead of hamsters. But we're going to look at what we mean by the P generation, the F1 generation, and the F2 generation. What we mean by phenotypic ratio, what we mean by genotypic ratio, and we're going to start going through some different patterns of inheritance. So let's start with complete dominance. And this story was figured out by Gregor Mendel. He used pea plants. I'm sure if you've taken a general biology course of any sort during junior high or high school or college, you've heard the story of the purple and the white pea plants. And he crossed purple flowers with white flowers and just determined that all the offspring were purple. I'm going to use a different example today. It's a very similar story though. So this is the story of an inheritance pattern called complete dominance. In the story of complete dominance, in this inheritance pattern, one allele is completely dominant over the other. So one allele is completely dominant over another allele. How do we know that? We look at the heterozygous individual. If in that heterozygous individual we see one genotype being expressed in the phenotype, then we know that was the dominant allele. So the heterozygous individual will have the dominant phenotype. It will look like whatever the dominant allele is. In other words, he has one of each allele, but he looks like the dominant one only. The way we look at ratios of offspring in each generation for each of these patterns of inheritance is we use what's called a Punnett square. And this Punnett square is just going to allow us on one part to look at possible eggs and one to look at possible sperm, and then we're going to do some fertilizations and see what our possible offspring are. So you need to understand something about meiosis in order to understand patterns of inheritance. So let's look at this first pattern of inheritance, complete dominance. We're going to represent the alleles using one letter. So rather than showing brown as a B and white as a W, we're going to use capital B and lowercase b. So we're going to have capital B represent the brown fur allele. And since we know that brown is dominant over white, we're going to have little b, lowercase b, represent the white fur allele. Usually what happens is whatever the dominant allele is determines the letter you're going to use. It really doesn't matter though. I could use the letter Z. It really doesn't matter. It just matters that the dominant allele is shown as a capital letter and the recessive allele is shown as a lowercase of that same letter, not two different letters. So let's assume that in this first generation, we have a brown dad, so obviously the brown male, and that brown male is going to mate with a white female. And we know that generation after generation, these brown bunnies have bred with other brown bunnies, and they've only had brown bunny babies. They've never had white bunny babies. So we know that this brown dad is homozygous dominant. In other words, for that brown fur allele, he has two brown fur alleles. So his genotype is going to be big B, big B, to show that he has both alleles the same, so he's homozygous, and they're both the dominant allele, which is brown, which we represent with a capital B. The white female, she can't have one of each, right? Or she wouldn't be a white female, she would be brown. So she's going to be homozygous recessive. And in the story of complete dominance, the only way you can have the recessive phenotype, the only way you can look like the recessive trait, is if you have two alleles for that recessive trait. Because remember, if she had one of them as a brown, she would look brown, not white. The only way she can be white is to be homozygous recessive. So her genotype is going to be little b, little b. So when they mate, we're going to use this Punnett square to look at what their possible offspring will be. And in order to do that, we draw a square and divide it in fourths. Remember that whether it's a male or a female, here's their chromosome number 18 that we pretended has that fur allele on it. Okay? And remember that when these go through meiosis, first thing they did was separate the homologous pair into two different cells, and now they're going to pull the sister chromatids apart. But by the time we're done, we're going to end up with 50% on average, 50% of the gametes are going to have one trait and 50% are going to have the other trait. Unless some get damaged along the way, whatever. But you're going to have 50% of your eggs having one allele and 50% having the other allele. If both your alleles are the same, it doesn't matter. We're going to split these. Remember, these separate. This homologous pair separated in meiosis 1. So half are going to end up with this one, half are going to end up with this one. They just happen to be the same. But we'll do our eggs on the top, and we'll do our sperm on the side. It doesn't matter where we do which one. So our only possible eggs, half are going to be little b, the other half are going to be little b. Possible sperm from dad, half are going to be big B, the other half are going to be big B. So when we do our crosses, we're going to draw a line and look at what the genotypes of our babies are going to be. This baby will be big B, big, little b, big, little, big, little, big, little. So all of our offspring are heterozygous. So offspring. The genotype of our offspring, 100% heterozygous. The phenotype, this is what determines that this is complete dominance. The phenotype is that all the offspring are brown. 100% brown. 100% brown fur. So in an individual that's an individual that's heterozygous, in a heterozygous individual, if they have the dominant phenotype, then we're looking at a story of complete dominance. This would be called the F1 generation. So F1 offspring, F1 generation offspring. Now let's take two of those offspring and cross them and see what we come up with. I know you don't really want to do that. You don't really want to have offspring mate with each other. Not a good idea. But in this case, we're just going to show if two heterozygous individuals mated. So, heterozygous mom and heterozygous dad. Let's look at that Punnett square. Same thing. These alleles split up when that homologous pair separates in meiosis 1. So half the gametes are going to be big B and half are going to be little b. Doesn't matter which parent we put on the top or on the side. Now let's look at our offspring. Big B, big B. So now our ratios are different. When two heterozygous individuals meet and mate, the offspring ratios are going to be different. We're going to have, for our genotype, I'll make a little chart, genotype and phenotype. These are our F2 offspring. This is the second generation. This is taking the offspring and crossing them. 25% homozygous dominant. What is their phenotype? They're going to be brown. 50% are heterozygous. Big B, little b. What color are they? They're also brown. And then 25% homozygous recessive. Those are our white babies. This type of inheritance is especially important in understanding human genetic disorders because most human genetic disorders are carried on the recessive allele. The only way you can have the disorder is if you're homozygous for the recessive allele. So when we're talking about a disorder that is called autosomal recessive inheritance, it means autosomal, that's telling you it's inherited on chromosomes 1 through 22, not sex chromosomes. Recessive means it's carried on the recessive allele. So that means the only individuals that have the disease are going to be these. So these individuals would have the disease or the disorder. Homozygous dominant, these are healthy normal. What are the heterozygous individuals called? They don't show symptoms of the disease, but they carry the allele for the disease. So they are called carriers. In the world of autosomal recessive inheritance, if we're talking about disorders or diseases, heterozygous individuals are called carriers. Carriers. This is significant when we're talking about human genetic disorders. Because if two carriers mate and they don't know that they're carriers because no one in their family has had an offspring, a child that has this disorder, it can be really bad. Because this is what will happen. This would be carrier mating with carrier. And on average, average, 25% of their babies are going to have the disorder. But that's assuming thousands of offspring. You're rolling the dice every time. Every time these two mate, every time they have an egg and sperm that come together, they have the possibility of having a baby that has the disorder. Because 50% of the sperm have the bad gene, and 50% of the eggs have the bad gene. So they're taking a bad roll of the genetic dice every time. There are many, many sad stories you'll hear of people who didn't know that they carried the disease allele until they married another carrier and they have a baby with this genetic disorder. And that's the only way they find out. So this is how many, many human genetic disorders are inherited. And by the way, a lot of those disorders just have one base on the DNA that is wrong. And that one base on the DNA results in the wrong amino acid in that spot, and that protein doesn't function correctly, and that baby, that person has a genetic disorder. That protein is not working right. Cystic fibrosis is an excellent example of this. Cystic fibrosis is inherited through autosomal recessive inheritance. And it is one base on the DNA that is wrong. And the babies who get both bad copies of that gene cannot make that protein correctly. They don't have a good recipe. If you got one good recipe, you're still making enough of that good protein. But if both recipes are bad, you can only make a bad protein. And in the case of cystic fibrosis, it's a membrane protein in the plasma membrane of those cells that allows salts to move in and out. And if it's misshapen, as it is in cystic fibrosis, those salts accumulate and mucus accumulates on the outside of the cells, especially bad in the lung cells. So one base wrong can result in a bad protein. If you have one good copy of the recipe in a heterozygous individual, you might be making enough good protein that you don't have symptoms of the disease, but you could pass it on to your offspring. So very important to understand this pattern of inheritance. So autosomal recessive means it's on chromosomes 1 through 22, and you inherit it on the recessive allele. The only way you have the full-blown disorder is if you have two bad copies of the recipe, two recessive alleles, homozygous recessive. So carriers are heterozygous in this type of inheritance. What happens though if we have a red flower and a white flower mate and the babies end up pink, not red? So we had brown and white mate and the babies were brown. But now we have red and white mate and the babies are pink. In that case, we're looking at something called incomplete dominance. And again, you need to look at the heterozygous individual to determine this pattern of inheritance. If that baby, if that offspring was red, then we would know we were looking at complete dominance. But those offspring have a phenotype that is intermediate. It's somewhere between the dominant and recessive phenotype. And that's what incomplete dominance is. In incomplete dominance, again, looking at the heterozygous individuals, the heterozygous individual has an intermediate phenotype. Heterozygous genotype equals intermediate phenotype. Again, the example is red flowers and white flowers. So if the red was big R, and the white was little r, all of our offspring are going to be heterozygous. So 100% heterozygous, and they're all pink. Oops. that would be an intermediate phenotype. In humans, we have a situation with cholesterol receptors that follows this pattern of inheritance. LDL and HDL cholesterol. If you remember receptor-mediated endocytosis, it was a situation where you have receptors on the plasma membrane that if certain molecules bind, they'll be brought into the cell and processed. And we have LDL receptors that bring in the bad cholesterol and break it down. 3 possible genotypes. I'm just going to represent them as big L, and then over here I'm going to have little l, little l. I'm going to make it loopy so you can tell it's an L. And then this would be our heterozygous individual. If you're big L, you have a lot of these receptors. We don't like these people. They can eat pizza and deep fried food and they never get high cholesterol. They're very good at processing this bad cholesterol. Little l though, these people have no receptors. These are people who usually by college age go in to get a physical and find out they have high cholesterol, and they end up on some type of medication to control their cholesterol at a pretty young age. If you're heterozygous though, which most of us probably are, you end up with something in between. You end up with an intermediate number of these receptors. So if you eat a healthy diet, you have low cholesterol, and if you eat an unhealthy diet, it'll be high. So intermediate phenotype in the heterozygous individual. That type of inheritance is called incomplete dominance.